This video highlights the GlobalRPh blog article by Similoluwa Oluwalana. Please like and subscribe to be notified of future videos from this writer. Diabetes is a chronic metabolic disorder primarily classified into two types, Type 1 and Type 2. Type 1, an autoimmune disease often diagnosed in adolescence, is marked by the body's inability to produce insulin. In contrast, Type 2, which constitutes the majority of diabetes cases, is associated with insulin resistance and is typically diagnosed in adulthood. By the year 2030, the global prevalence of Type 2 is expected to rise, primarily due to unhealthy lifestyle choices such as poor diet and lack of physical activity, leading to obesity. Insulin, a protein hormone, is crucial for diabetes management. While subcutaneous injections are the most common delivery method, they have limitations, including poor patient compliance and rapid insulin clearance. Non-invasive delivery methods, particularly oral administration, have been explored extensively but remain hindered by challenges related to insulin bioavailability in the gastrointestinal tract. Despite promising developments, oral insulin has yet to reach clinical application. Research into oral insulin delivery has faced numerous challenges since the first attempts in 1921, shortly after insulin's discovery. Early attempts at getting a viable delivery system, like giving patients insulin enterally or changing formulations by adding alcohol, did not lead to a better metabolic state. Despite setbacks, companies like Emisphere, Diabetology, and Oramed continued to develop oral insulin formulations, with clinical trials from 2001 to 2019 showing mixed results. Often, trials failed to demonstrate superior glycemic control over placebo, largely due to small sample sizes and challenges with insulin bioavailability. The gastrointestinal environment, with fluctuating pH levels and digestive enzymes, degrades insulin. The stomach's low pH and enzymes like pepsin and trypsin cleave insulin into inactive peptides. Formulations like Oramed's ORMD0801 use proteinase inhibitors, but long-term concerns remain. Researchers have explored mucosal adhesion systems using materials like chitosan to enhance absorption. The epithelial barrier and tight junctions also limit insulin absorption. Strategies using cell-penetrating peptides have shown modest improvements, but bioavailability remains low. Recently, experts have started investigating new oral insulin delivery systems, focusing on the encapsulation of insulin within different types of nanoparticles to enhance its stability and absorption in the gastrointestinal tract. These systems were tested in preclinical models using animals like rats to assess their effectiveness in improving insulin bioavailability. Different formulations were explored, including lipid-based nanoparticles, such as solid lipid nanoparticles and nanostructured lipid carriers, polymeric nanoparticles like chitosan-based systems and hydrogels, and inorganic nanoparticles, such as mesoporous silica and quantum dots. The studies assessed the ability of these carriers to protect insulin from degradation by stomach acid and enzymes, as well as their efficiency in facilitating insulin transport across the intestinal barrier. Key parameters evaluated included insulin encapsulation efficiency, bioavailability rates, and the impact on blood glucose levels in diabetic animals. Additionally, safety assessments were conducted in some cases, examining the potential toxicity of these carriers over long-term use. Many nanocarrier-based oral insulin systems are currently being explored. Nanocarriers are tiny particles, often under 1,000 nanometers in size, designed to protect insulin from degradation in the gastrointestinal tract and improve its absorption. These carriers can be classified into lipid-based, polymeric, and inorganic nanoparticles. Lipid-based nanoparticles, like solid lipid nanoparticles and nanostructured lipid carriers, are advantageous for cellular uptake due to their lipid composition. Still, they face challenges with encapsulating hydrophilic molecules like insulin. Despite these challenges, some solid lipid nanoparticle formulations have achieved bioavailability rates of around 5% in animal studies. Polymeric nanoparticles, another important category, use natural or synthetic polymers such as chitosan and lactic coglycolic acid. The FDA has approved these polymers for their biocompatibility. Chitosan-based formulations, for instance, have shown promise due to their ability to interact with the mucus layer in the intestines and improve insulin absorption, achieving bioavailability rates of up to 18% in animal models. Insulin can also be encapsulated with hydrogels and micelles. It provides sustained release and enhanced stability in the gastrointestinal tract. Inorganic nanoparticles, such as mesoporous silica nanoparticles and quantum dots, are also gaining traction in oral insulin delivery. These carriers offer exceptional drug-loading capacities and protection for insulin in the harsh gastrointestinal environment. For example, mesoporous silica nanoparticles, modified with polyethylene glycol, have been reported to stabilize insulin and improve its bioavailability. However, concerns about the long-term toxicity of inorganic nanoparticles remain, particularly regarding their effects on the liver, kidneys, and nervous system. Surface modifications of nanoparticles have been explored to enhance the stability and absorption of oral insulin. Various strategies include using coatings such as hydrogels and gelatin, which protect insulin in acidic environments. Hydrogels, for instance, have demonstrated the ability to control insulin release in diabetic mice, while gelatin-coated nanoparticles maintain stability in gastric conditions. Additionally, pollen encapsulation techniques have been shown to significantly improve oral insulin bioavailability, leveraging the protective structure of pollen particles. Adjusting the surface charge of nanoparticles has also been investigated. Negatively charged nanoparticles enhance mucus permeability, while positively charged ones prolong their presence in the mucus layer. Interestingly, studies have shown that surface charge only has a limited impact on insulin bioavailability, as negatively and positively charged nanoparticles exhibited similar absorption rates in diabetic rats. Lastly, zwitterionic and protein-based modifications further improve nanoparticle permeability and stability, highlighting the importance of surface characteristics in oral insulin delivery systems. Several clinical trials have assessed the efficacy of different oral insulin formulations. Nodlin trademark, developed by Nod Pharmaceuticals, was tested in a Phase 1 clinical trial in 2012 with 12 healthy volunteers. It showed promising glucose-lowering effects but had high variability in efficacy. HDVI by Diasum Pharmaceuticals targeted liver cells and showed superior hypoglycemic effects in a small trial. A larger Phase 2-3 study began in 2008, but results are still pending. Oramed's ORMD0801 showed promise in Phase 2, but failed in Phase 3 with no significant improvement in glycemic control. Overall, while initial trials have shown potential, the success of oral insulin in clinical settings has been limited. Further research and refinement of delivery systems are needed to overcome current challenges. Although significant progress has been made in developing oral insulin, several challenges remain before it can become a viable treatment. Initial trials, such as those involving Nodlin trademark and HDVI, showed some promise in lowering blood glucose levels. Still, issues like variability in efficacy and incomplete data from large-scale studies hinder conclusive results. Despite encouraging Phase 2 outcomes, Oramed's ORMD0801 failed to demonstrate significant improvement in glycemic control in a recent Phase 3 trial, highlighting the difficulties in achieving consistent efficacy. While some formulations have advanced to later stage trials, reliable and effective oral insulin treatments are still not ready for widespread use. The study concludes that significant clinical success remains elusive despite extensive research and innovative approaches to develop oral insulin, such as nanocarrier systems, oral microneedles, and gene therapy. While certain strategies have shown potential in targeting specific cells and improving oral bioavailability, clinical trials have yet to produce effective results. The failure of promising candidates in advanced trials underscores the ongoing challenges in creating a viable oral insulin delivery system, making it clear that more research is needed before it can become a practical treatment option for diabetes. If you enjoyed this video, please like and subscribe. Subscribing also helps ensure you never miss a new video release from GlobalRPh.